Hello students, in this part, or part 2 of the circulatory system, we're going to be talking about cardiac conduction. We'll understand a little bit more about the EKG and those different waves. We'll also talk about cardiac cycle. So we'll go ahead and start. First off, we'll talk about cardiac communication, which means how do the cardiac cells communicate together since we need the heart to pump as a unit. We can't have, some cells of the atria contracting at one time and then the other part of the atria contracts at a different time. And that's, that is just going to lead to a whole mess in the heart. So in order for it to function as a unit, there has to be a fast way of these cells to communicate together. And we have about 1% of cardiac cells do not function in contraction. So they're not muscle cells. They are differentiated into neuro cells, okay, where they have the ability to become pacemakers. They're kind of like batteries for the heart, and they're known as the cardiac conduction. It's kind of like the wiring of the heart. And these cells, together with the cardiac muscles, they're all connected through these gap junctions, and that facilitates the fast passage of the action potential through all of the myocytes. Okay, so we'll take a look at the conduction system. You'll find that the wiring is found in yellow. In our cells, when you actually dissect a heart, you're not going to really find wires. Okay, you're not going to find cells that look different. On the gross picture, by the naked eye, you really don't see any difference. But they're kind of highlighted here in yellow, where we have the SA node or the sinoatrial node. It's found in the upper part of the right atrium, right before or right by where the superior vena cava kind of enters into the right atria. And then we have these fibers that are going to spread the action potential to both atria. So you see here the right atria, and then you have that little branch kind of spreading it down over to the left atria. And then electricity would have to go through an atrioventricular node, which is the AV node, and then to the bundle of His. His is the scientist that discovered it, also known as the AV bundle. And that bundle will go down into the interventricular septum, which is the septum between the two ventricles, and it will branch out into a right and a left branch. Now, these branches are going to go down on the corresponding ventricular walls and then reflect on the ventricular wall and give out other smaller fibers, and those smaller fibers are known as Purkinje fibers. And that is how the electricity spreads. The action potential in these wires can only go from atria down to ventricles. It does not have the ability to go backwards. So if there is an ectopic focus, starting electricity down here in the ventricles, it has no way of going back into the atria. So the excitation of the heart, our pacemaker of the heart, is the sinoatrial node, or the SA node. And as we mentioned, it's found in the right atrium. And then it's going to spread that action potential down to both atria and then down to the AV node. There is a little delay, at the AV node. That delay is about 0.1 of a second. And that allows for the atria to contract and give a little delay before the action potential goes down to the ventricles, leading to their contraction. You do not want the atria and the ventricles to be contracting at the same time, because then that is going to prevent the ventricles from filling up with blood. You want the atria to contract while the ventricles are relaxing, and during that delay, the ventricles are going to be relaxing and gathering the blood from the atria. And then the ventricles will contract. So this 0.1 second delay has is very important clinically. Well, and then we mentioned it going through the AV bundle, then down to the bundle of His, and then down to the two bundle branches, a right and a left. It'll descend in their corresponding ventricles, and then it'll reflect and give out smaller branches, which are your Purkinje branches. And that is how the electricity spreads out through the ventricular walls. So you can see here the sequence of cardiac excitation. So first off, we have the atrium contraction. or the atrial excitation, where the SA node is going to spread out through the what are known as the atrial syncytium or the atrial unit, and then down to the AV bundle. Okay, so this is where the atrium contracts. Both atria contract at the same time, and that is going to show up on the EKG or the electrocardiogram as this little tiny wave known as the P wave. And then you have a 0.1 second delay at the AV junction, and you can see here through what is known as your PR interval on the EKG, and then the electricity will go down the bundle of His and the branches and then the Purkinje fibers. That'll lead to ventricular contraction. Ventricular contraction is going to be shown on an EKG, will be recorded as this complex known as the QRS complex. And then when the ventricles relax, you'll record that electricity as this final wave known as the T wave. Okay, T as in Tom. So you want to be able to correlate the different waves with the what's going on in the heart. So again, your P wave is atrial contraction, P as in Paul. Then you have this QRS complex, that is your ventricular contraction. And the last T as in Tom, T wave, that is your ventricular diastole or ventricular relaxation. We do not see an EKG wave when it comes to atrial diastole or atrial relaxation because it happens at the same time as ventricular contraction. So it's really masked by this huge QRS complex. And then again, don't remember that PR interval, and that represents the delay of the action potential as it goes from the atria down to the ventricles. So what happens in the ventricular muscle cell? So just like any other muscle cell, there is a membrane potential. So this graph shows you the potential of a ventricular muscle fiber. The resting membrane potential in ventricles is at a negative 90 millivolts. Okay, and remember, generally speaking, we said that depolarization happens due to entrance of sodium, while repolarization is due to the exit of potassium from the cells. So if you might want to go back and kind of review that when we talked about skeletal muscle fibers. So those rules do apply on ventricular muscles. But you are going to notice this little plateau right here. So this plateau represents where you have at the same time, you have potassium exiting out of the cell while calcium is entering at the same time. So you have a positive ion leaving and another positive ion coming in. And that leads to very minimal changes in the membrane potential. And that plateau is important in the ventricular muscles because this is when the ventricles are relaxing and they are filling up with blood in order to fill up as much as possible with blood. Okay, the calcium channels that are here are known as L channels. Also, the L stands for long-lasting or DHP channels. Now for the nodal cells, which are the SA node and the AV nodes, those have what is known as a pacemaker potential. Pacemaker potential means that it has the ability to produce electricity without an order from the brain. So even after brain death, now there's no orders coming out of the brain, the heart still has the ability to pump for a while, very limited time, and that is due to the pacemaker potential of your SA nodes and the other cells like the AV nodes and the Purkinje fibers. The SA node, though, is known as the pacemaker because it has the fastest rate of automaticity. And automaticity means that, again, the capacity of the spontaneous rhythmic self-excitation. Again, it is a specialized kind of cell where it has the ability to produce its own electricity without orders coming from the brain, from higher centers in the CNS. And because the SA node is the fastest at doing this, then the upper hand is to the SA node, making it the pacemaker of the heart. If the SA node, though, is damaged for whatever reason, we have other parts that can take over. So we have the AV node, for example, the bundle of His, or the Purkinje fibers. All of these, again, have that automaticity capacity, but they are at slower rates. And that's why they don't get the upper hand unless this faster one is damaged. Now we want to talk about the membrane potential in the cardiac node. So at these nodal cells that we talked about, the SA node or AV node or Purkinje fibers, how is it that they have that ability to produce their own energy? Well, first off, there is no resting membrane potential. Okay, so there is not a time where these nodes are at rest. They are always continuously changing their potential. So that is an important thing, and that gives them that automaticity characteristic. So there is no resting membrane potential. When it's in the negative territory, okay, we have what are known as F-gated sodium channels. And F actually stands for funny gated sodium channels, meaning that they are pretty much always open. So, what's the point of having a gate if it's always going to be open? That hence the name funny gated sodium channels, okay, because it's kind of ironic that there's a gate, but we don't shut it. Okay, so anyway, so these F-gated sodium channels, they allow for the entrance of sodium, okay, and that is going to help in the depolarization. We also have calcium entering into the cell. Okay, when it reaches the threshold potential, the sodium channels are going to shut and only calcium is going to be allowed in, okay? And so that is, the calcium entrance is what finishes off depolarization. At the tip of this, your calcium channels shut down, again, those L calcium channels, and we open up potassium channels in order to repolarize back into negative territory. For a little bit on conduction disorders, so what happens, we kind of already talked about if SA node is damaged for whatever reason, the other parts are going to pick up, like the AV node, the bundle of His or Purkinje fibers. Again, they have slower rates, but they're good enough to keep the person alive. And, okay, in order to treat an AV conduction disorder, we need to put in an artificial pacemaker, okay, and that is going to stimulate the ventricular cell at normal rate. I mean, even though there might be that inherent characteristic of automaticity in the ventricular Purkinje fibers, the rate is too slow. Okay, so sometimes it can reach like 20 or 30 per minute, which are really, really slower rates. Again, maybe enough to keep them alive, but not enough to function normally. So you want to put an artificial pacemaker in order to contract the ventricular muscles at the normal rate. So looking at an electrocardiogram or an ECG or an EKG, okay, that is how we record the electrical events that are happening in the heart. Know that we're not really, because we don't have the ability to put those electrodes directly onto the cardiac cells, we are actually recording what is happening in the extracellular fluid surrounding the muscle cells. So again, it's not a direct recording of the membrane potential, but it gives us a very good idea of what's going on. And we have all of these leads, which are basically the electrodes. They're put on different parts of the chest and different parts on different limbs. We have different recordings, but we're going to talk about the one that gives us, better understanding or a more clear understanding of what's going on. And this is where we put the electrodes on the wrists. Okay, so it'll give us this, what is kind of known as the default kind of EKG. So we talked about the different waves. We have a P wave. Again, that stands for atrial contraction or atrial systole. We have this PR interval that gives us a good idea about how long it took for the action potential to go from the atrium to the ventricle. QRS complex represents ventricular contraction or ventricular systole. And this T wave represents ventricular diastole or ventricular relaxation. Again, we do not record what's happening during atrial diastole because it occurs at the same time as ventricular contraction and it is masked by this huge QRS complex. So this is what we talked about. So the P wave is atrial depolarization, which corresponds mechanically to atrial contraction. QRS represents ventricular depolarization, which corresponds to ventricular contraction. And the T wave shows us is the recording of ventricular repolarization and corresponds to ventricular diastole. And as I said, atrial repolarization is not really recorded because it's masked by the QRS. Now for the excitation contraction coupling. So what happens, what is the role of calcium when it comes to the contraction of muscle fibers? So there's a little difference between muscle contraction in skeletal muscles and muscle contraction in cardiac muscles. And when we talked about skeletal muscles, we said that calcium is going to attach to all of the available troponin. And that's not the case in ventricles, or not in the case, in cardiac muscle fibers. In cardiac muscle fibers, calcium is going to attach only to some troponin, and it's going to leave some untouched. So that if we need the heart to contract harder or faster, we have a reserve of troponin to attach to in order for that to happen. So your skeletal muscle fibers, we have that all or none rule. Either contracting the muscle fiber or you're not. That's a little bit different than cardiac muscle fibers. So we have the ability to contract the heart at a, normal rate. We also have the ability to contract it at a faster and harder rate, say during exercise. Okay, so calcium, you have to have a little bit reserve of troponin available so that when we exercise, the heart can go faster and stronger. So, and that is, one of the main differences between the excitation contraction coupling in cardiac muscles as opposed to skeletal muscle fibers. So if you look here, at this diagram that by now I'm sure gives us all nightmares, including myself. But what happens is that on our cardiac muscle fibers, we have these beta adrenergic receptors. Beta adrenergic receptors are going to be stimulated by either norepinephrine or epinephrine. That is going to lead to the activation of adenylyl cyclase enzyme, increasing cyclic AMP inside of our cardiac muscle cells. When that happens, okay, that is going to open up these L-type calcium channels. Calcium goes into the cardiac muscle cells, attaches to these ryanodine receptors, okay, that are found in the sarcoplasmic reticulum. Now calcium is going to be released from the sarcoplasmic reticulum, and then calcium will start attaching to some of the troponin, leading to that sliding of the actin on myosin, okay? So again, you can kind of go back to the skeletal muscle fiber contraction in order, to if you feel the need that you have to go ahead and do that. The main difference in this whole story is that calcium does not attach, in this case, to all of the available troponin. You want to have kind of a reserve. Okay, now for a cardiac cycle. So there's a continuous cycle of contraction of the heart. The two atria on top contract at the same time. And while that is happening, the two ventricles on the bottom are in relaxation. And then this switches, where the atria on top relax and the two ventricles on the bottom are contracting. There is a video on that I posted on Moodle, and it's somewhere in these slides as well. So I would definitely recommend that you take a look at it. It shows you in animation the whole cardiac cycle events. So again, when we talk about the cardiac cycle, now we're going to be talking about it in terms of ventricles. So when we say systole and diastole, we by default mean ventricular systole and ventricular diastole, even though we're not talking about ventricles. I don't specifically say ventricular. Our heart beats, on average it's 60 to 90 per minute, but on average at rest, it's about 72 beats per minute. If each cardiac cycle lasts for less than a minute, so 0.8, you'll see that most of the cardiac cycle is in diastole, so 0.3 seconds, while the remaining 0.5 seconds is found in diastole, meaning that you want the ventricles to relax in order to fill up with blood, okay, while the contraction part, the systole part, only takes less of that cardiac cycle. So the cardiac cycle means one contraction followed by one relaxation, one systole followed by one diastole. That is your whole cardiac cycle. The majority of that cycle is actually occurring, more than 50% of it would be in diastole. So you want to fill up those ventricles in order to contract. So this gives you an idea of the cardiac cycle. We start off, remember, this is a cycle, so you can really start off anywhere in this cycle. So this is the part, that we'll go with what's in the textbook. So it starts off at what is known as isovolumetric ventricular contraction. So we'll break it up. Ventricular contraction means that the ventricles are going to start to contract. Isovolumetric means there is no change. Iso means the same. Volume. There is no change in volume in the ventricles at this point when they start the contraction. So we'll understand why that is. During isovolumetric ventricular contraction, all four valves are shut. Okay, so you'll see here that the AV valves are closed, and so are the semilunar valves. Those are closed as well. The ventricles are going to start the contraction with all valves closed. And that again is your isovolumetric ventricular contraction. The pressure inside of these ventricles is going to increase. When it gets higher than the pressure found in the aorta or the pressure found in the pulmonary trunk, that is going to open up these semilunar valves. So you'll see in the part known as ventricular ejection, opening up of the semilunar valves while the mitral and the tricuspid are still shut. Okay, so now these ventricles contract, it will push blood into the aorta and into the pulmonary trunk. So your ventricular systole is made out of two different parts, the isovolumetric ventricular contraction with all valves closed, and the ventricular ejection phase with the semilunar valves open. That'll take us down to diastole, again, of the ventricles. Okay, the first one would be isovolumetric ventricular relaxation. So you can guess isovolumetric means that there is no change in volume, which means that all of the valves are shut. Okay, so again, your AV valves and semilunar valves, all four of those valves are closed. Whenever you see the term isovolumetric, whatever, that make that kind of like a reflex that all of the valves are closed. Okay, followed by the phase of ventricular filling. When the ventricles relax, okay, that is going to lead to the pressure in the atrium up here to become higher than the pressure in the ventricles. That will lead to the opening up of these AV valves. Now, the first part of ventricular filling is more of a passive event where by gravity, the blood is going to flow from the atria down into both ventricles. And then the last part of ventricular filling would be where the atria actually contracts. Okay, and these valves are still obviously open, and that will lead to the more filling of these ventricles. Okay, again, I would definitely recommend, again, going back to the video, listening to it, and maybe if you still, if you have to go over this again, feel free to do so. So what is going on? This is the video that I was referring to. Now we want to kind of go over what are the changes in pressure that are happening in the right ventricle and in the pulmonary artery during the cardiac cycle. Okay, so the green one represents the pressure in the pulmonary artery, and the orange curve represents the changes in pressure in the right ventricle. So this is something that you really kind of want to maybe understand as opposed to having to, memorize this. So we'll start off kind of just like we started off in the previous slide. We'll start off with the isovolumetric ventricular contraction, which is this phase right here. So isovolumetric ventricular contraction means the ventricles are contracting while all valves are closed. Okay, so try to think about it. What do you think would happen in the pressure in the right ventricle? Well, that is going to definitely increase. Okay, while the pressure in the pulmonary artery is going to be getting less because there's no blood being pushed into it yet. And then the phase of ventricular ejection, that's when the pulmonary valve opens and the ventricles contract. So you'll see here that orange graph, the ventricular pressure increases, pushing blood into the pulmonary artery. So you'll see here that the pressure in the pulmonary artery increases as well. And then stage 4, which is isovolumetric relaxation, where the ventricles start to relax and all of the valves shut down. So you'll see the pressure going down in the ventricles. Same thing with the pulmonary artery. Pulmonary artery, the blood that was just pushed into it from the right ventricle is going to be moving further into the lungs. Okay, so it's going to be emptying gradually, and you can see that gradual decrease in pressure until we reach the isovolumetric ventricular relaxation, number 1. The ventricular filling portion, which now we'll open up the AV valves, and blood is going to start entering into the right ventricle, and then the atrial contraction happens and fills it up again even more. So see here that little bump that represents atrial contraction filling up the right ventricle. Now, a little on clinical application. If you've used a stethoscope in order to listen to a normal heart, we have 2 different heart sounds. There's S1 and S2. S stands for sound. Okay, so the first heart sound, which is a very low-pitched lub sound, and that occurs due to the closure of the AV valves. Okay, so the AV valves, as they shut, that will give you that first heart sound. The second heart sound, which is a little bit louder, kind of sounds like a dub sound, that occurs due to the closure of the semilunar valves, so the pulmonary and the aortic valves. These are your normal heart sounds, but we have some abnormal heart sounds, okay, and we call those heart murmurs. Sometimes they are a sign of heart disease. In some individuals, it's considered their normal. Okay, so, that is definitely, has to be evaluated. But on general, if the blood or if there's any kind of vibration, abnormal vibration when the valves shut, that can lead to a heart murmur. And like I said, in a good number of the population, that is normal for them. As blood passes through the valves, that passage is very smooth. Okay, so we don't hear blood passing through the different valves, healthy valves. That is known as laminar flow. There's no turbulence, we don't hear it. But if turbulence does occur for whatever reason, if the valves are too narrow, like in stenosis, for example, or if the valves start leaking, like in insufficiency or regurgitation, that is going to lead to turbulence in the blood flow, and now we can start hearing it as a heart murmur. So you can see here, these up here represents a normal open valve. The blood flowing in a laminar flow, it's quiet, we do not hear any murmurs. This represents a stenotic valve. So this is a valve that is narrow. You can see the turbulence of the blood. Now we can hear that turbulence in the form of a murmur. As the valve shuts, okay, again, the blood is now being kind of pushed away from the valve in a linear laminar flow. Again, that's quiet, no sounds. But if there is an incompetent valve or an insufficiency in the valve, you'll see blood is regurgitating back where it shouldn't be. And that leaking is a turbulence flow, and that leads to a murmur too. Now we'll start talking about, a little bit of the mechanics of the heart. So cardiac output, okay, from the name, it tells you that a cardiac output is how much blood, or what's the volume of blood that is pumped out of the heart each minute. And you can calculate it by finding out the heart rate per minute and multiplying it by the stroke volume. So the stroke volume means what is the volume of blood that is being pushed out of the ventricle with each ventricular contraction. Okay, so that again is your stroke volume. If you multiply it by the heart rate that will give you your cardiac output. Your normal cardiac output is about 5 liters of heart per minute. Obviously, it varies from one person to another, but this is on average at rest what it is. Okay. If for whatever reason, things change, like the heart rate goes up or the stroke volume goes up or the stroke volume decreases, that is going to change, could potentially change your cardiac output. So we'll take a scenario, let's say if somebody is bleeding, okay, that'll definitely decrease their stroke volume. So in order to keep the cardiac output constant, the heart rate would have to increase. So if somebody bleeds, to keep up with the function of the heart, now that the stroke volume is less, the heart is going to have to pump faster. So there is an increase, there's a tachycardia, a reflex tachycardia that happens due to the loss or decrease in stroke volume. Now, how does the heart regulate itself? Okay, so normally at rest, the SA node fires up at about 60 to 90 per minute. It could be slower, it could be faster, and during the day, there is an adjustment needed. So when we're at rest, when we're sleeping, it could be slower, but when we're awake and walking or exercises, it has to pump a little bit faster. So how does the heart do all of this regulation? Well, there are a couple of different things. Okay, obviously the heart has an autonomic nervous supply. So there's parasympathetic and sympathetic fibers. The parasympathetic fibers comes from the vagus, and the sympathetic fibers are just different sympathetic nerve fibers. Okay, your parasympathetic is going to inhibit the heart, so it'll decrease heart rate, it'll decrease ventricular contraction, it'll decrease the heart rate, and that is pretty much it. The sympathetic stimulation, though, will lead to an increase in heart rate and an increase in the strength of ventricular contraction. Okay, these effects are known as chronotropic. So chronology means kind of like time-wise. Okay, so the chronotropic effects of para and sympathetic would be parasympathetic again, would lead to decrease the heart rate, while sympathetic will stimulate the heart rate. You can see here the different effects of sympathetic versus parasympathetic. The sympathetic nerve fibers use both norepinephrine or epinephrine, and they stimulate these beta adrenergic receptors. Sympathetic receptors are found on the SA node, AV node, on the atria, and in the ventricles. So pretty much we have beta adrenergic receptors on all of the parts of the heart. So on the nodes, it'll stimulate both of them. So it'll increase your heart rate due to stimulation of the SA node. It stimulates the AV node, so now these action potentials will go faster, will reach the ventricles faster, so it increases the conduction rate at the AV junction. It increases the force of contraction of the atrial and the ventricular muscle fibers, so the heart is going to be pumping faster and stronger. When you compare that with the parasympathetic fibers use acetylcholine, and the heart has muscarinic receptors. Muscarinic receptors are found in the SA node, AV node, and atria, but it is not found on the ventricles. Okay, so there is a very limited effect of the parasympathetic nervous system when it comes to ventricular contraction. Parasympathetic fibers are going to decrease heart rate due to their inhibition of the SA node. They're going to decrease the rate of conduction through the AV bundle, and it'll decrease the force of contraction of the atria. And again, it has a non-significant effect on the ventricular force of contraction. Now, the stroke volume, which we kind of already discussed, is how much blood, or the volume of blood, that is pumped out of the ventricle with each ventricular contraction. And it is the difference between what is known as the end diastolic volume and the end systolic volume. So the end diastolic volume means when the ventricles relax during diastole, how much blood would be in it. Okay, so your end diastolic volume is the volume of blood in the ventricles at the end of its diastole. while the end systolic volume means at the end of systole, at the end of ventricular contraction, how much blood is left inside of the ventricles. When you subtract those two from each other, it'll give you about what is known as your stroke volume, which is about 70 milliliters per beat. So with each stroke of the ventricles, with each ventricular contraction, it is pushing out about 70 milliliters of blood forwards. And there are three different factors that are important to that affect the stroke volume. Well, since this is what this is the equation, so obviously your end diastolic volume is definitely one of them, and the end diastolic volume can increase if the venous return is increased. Okay, your venous return means how much blood returned to the right atrium. The more blood that is returned to the heart, the bigger that end diastolic volume, which means, which will have an effect on the ventricles. This is known as the preload. Okay, so the preload again is your venous return. Any change in the afterload would be, now that the ventricles have to contract, it has to contract against a pressure. So that pressure could be in the aorta, in the case of the left ventricle. So how high or low the pressure in the aorta is, that is what your afterload would be. Okay, so that is the arterial pressures against which the ventricles have to pump. And then any change in the magnitude of the sympathetic nervous system to your ventricles. So all these three, end diastolic volume, which is basically a function of the preload, and then any changes in afterload, so any changes in blood pressure, and then the effect of the sympathetic nervous system, all three of those have an effect on the stroke volume. Now there is a law of the heart known as Starlings Law of the heart that is related to Frank-Starling mechanism. Frank and Starling, basically what they say is it explains the relationship between the end diastolic volume and the stroke volume. And what it says is that the more you stretch a cardiac muscle, the stronger the contraction. Okay, so kind of, imagine this as a rubber band. When you stretch it further, and you let go, it'll snap harder. And that is what the ventricles do. Okay, if you stretch the ventricles more by increasing the end diastolic volume, that is going to lead to a stronger ventricular contraction. Up to a limit though. Okay, so there's a limit. If you overstretch the ventricles, that could lead to heart failure. So there's definitely a limit to this law. Okay, so again, any increase in your venous return. Okay, so for example, exercise. When you exercise and you move your skeletal muscles, that will increase your venous return to the heart. That is going to stretch the ventricles because now it's filling more and lead to a stronger contraction. Okay, again, this has its limitations. So here you can see this is at normal resting value. Okay, and this is your ventricular end diastolic volume, and this represents your stroke volume. As we start exercising, venous return increases, and your ventricular end diastolic increases as well. So you'll see here that your stroke volume, force of contraction will increase, leading to the increase in stroke volume. But there is a limit to this. It's not a linear relationship. There is a limit where this curve is going to, no matter how much you've overstretched the cardiac muscles, and that could lead to ventricular failure. So how is it that the sympathetic nervous system has an effect on our cardiac muscle fibers? It's all due to the effect on the adenylyl cyclase enzyme. Remember the norepinephrine/epinephrine, when they connect or when they attach to the beta adrenergic receptors, they activate adenylyl cyclase, and that leads to the increase of intracellular cAMP or cyclic AMP. And that is your effect that is going to occur. So it's all due to the stimulation of cAMP. That is going to lead to more increase in intracellular calcium, Now we can use that reserve of troponin that we kind of left off when we are at rest and in order to pump faster and stronger. Now, what is the ejection fraction? Ejection fraction means it's a ratio between your stroke volume and the end diastolic volume, meaning that your ventricles, let's say these are, I'm making these numbers up. So your ventricle at the end of diastole, let's say, has about 100 milliliters of blood, and then your stroke volume, when it contracted, it pumped out, say, 10 milliliters. So your stroke volume would be 10 over 100, which is about 10%, which means that your heart is, the heart in this condition, in this example, is not really a well-functioning heart. So your ejection fraction is expresses, the ejection fraction is expressed as a percentage. Okay, so you'll see like 50% or 75%. This is the percent, that's your normal ejection fraction in a healthy individual. Any increase in ventricular contraction is going to increase the ejection fraction. Now, what is the effect of the afterload on stroke volume? So again, your afterload is pretty much indicates how hard the ventricles would have to contract in order to overcome that pressure behind, in front of it. So if we're talking about the left ventricle, what is the pressure in the aorta, and how hard would the ventricles have to contract in order to overcome that pressure? So any increase in the arterial pressure is going to reduce your stroke volume, okay? That's because, for example, when you're trying to carry something with a skeletal muscle, the heavier it is, okay, that increase in load is going to make it harder for your muscles to carry it. The same thing with with your cardiac muscles. If there's an increase in blood pressure, that's going to make it harder for the ventricles to contract. So any increase in afterload, that means the heart has to work harder in order to overcome it. Now, this is the end of the second portion, and on to part 3.